práctica tipo de células.
Good day, teacher Diana Duque, another college. In this video is fulfilled the purpose of demonstrating the laboratory practice number 1 on the 17th of March of the subject, cellular and molecular biology. Focus on cell types and cellular structure. This practice was performed by the following students of the course 402: Santiago Buitrago, Correa Pedro, Estrella Miller, Herrera Mariana, and Gabriela Leiva of the University Cundinamarca of the Agronomy Engineering Program. However, before starting the observation of the cell purpose in the practice, it is case to give an introduction to the subject. The study of the cells and their structure is fundamental to understand the basic principles of biology. Cells, as we have already studied in previous sessions, are the structural and functional unit of all living beings, and can be classified into two large groups, prokaryote cells and eukaryote cells. Within the eukaryote cells, the famous animal and plant cells are differentiated, each with particular characteristics that allow them to fulfill various functions in organisms. The main idea of the course of the video is to establish the structural and functional characteristics of cells from scientific classification criteria between different phylogenetic groups. This classification will be carried out with the use of optical microscopes with different views objectives, identifying their main structure and differentiating their morphological characteristics. Methodology of the practice. The laboratory practice was carried on in order of the following observations. The first one was observations of prokaryote cells, protozoa microalgae with the help of a sample of current rested water or rice. The second one of an observation of eukaryote cells with a sample of yeast. The third observation focused on epithelial cells with a sample of the lower part of a cheek. The fourth observation of leaves of an Elodea plant. The fifth of the observation of chromoplants with tomato pulp. The sixth observation focused on chromoplants with samples of the tree plants material of different species. And the 17th and last one observation of a potato cut. Next, we will proceed to the analysis and procedures of this laboratory practice.
General objective: to observe and describe the morphological characteristics of prokaryote and eukaryotic cells through microscopic observation of different samples. Specific objectives: to identify the cellular structures present in eukaryotic cells of different plant species. Document observation of prokaryotic and eukaryotic cells by means of photographies or other graphic records, putting out their components.
Observation of prokaryotic cells, protozoa, and microalgae. A small amount of water sample was extracted from a well using a pipette. This small amount was placed on a slate and then covered with a cover slip. The slate was then placed under a microscope for observation and 10x magnification. This view shows microalgae. Microalgae are spherical dots with a very intense green color due to the presence of chlorophyll. They also have a softly very thin wall. This view shows lay protozoa moving rapidly throughout the water due to the action of cilia. These are oval-shaped ciliates, more especially those of Paramecia. They are translucent and vacuoles, and nuclei can be seen inside. Some rootlets were also observed. The same procedure was repeated, but this time neutral red was applied to kill all living organisms in the samples, so it could be viewed under a microscope at 10x magnification. Here, a purple protozoan was identified, which did not move because it was dead.
Next, I will guide why to the observation number 2 of eukaryote cells. Based on a sample of yeast, its procedure was as follows. In the letter of warm water with sugar, a carbon source for jets. In a beaker was dissolved and pinch of jets, as seen in the video. The sample was left to incube for 5 minutes. Then on a slide was added a drop of the previous solution of jets. Then in the video is observed under the microscope with the help of a lens with objective of 40x, the shape and size of oval and spherical cells of small size of a color bedouin wishing a transparent with cell walls and different from each other. These are different texture walls or cell membranes. These divide their groupings, which may indicate a state of active growth. The two not present are apparent mobility. In this case, there were no bubbles or clear space in the microscope, since this, if presented, could indicate the beginning of the fermentation process with the production of gas bubble CO2, indicating metabolic activity, which does not correspond to the was observed in the yeast practice. It should be noted that the yeast are carried unicellular fungi, commonly used in fermentation, and the species is for more frequently than we think, aiding in break or beer. Subsequently, a drop of methylene blue was added to the preparation, a vital dye used in laboratories for the differentiation of live and dead cells. Useful for evaluation cell biology, highlighting cellular structures, studying the nucleus and cytoplasm in eukaryotic cells for better visualization under the optical lens of for a microbiological units, using samples stained to different microorganisms in what is labeled culture of tissues. In this case, a drop of stain was added to the cover slip without forming bubbles, thus accommodating the slide of the microscope, and with a 10 and 100x objective. We use this dye to different viable cells from non-viable cells, giving the mechanism action the stain blue indicating to the they are dead or damaged. This occurs because methylene blue cannot cross intact cell membranes, but it does enter where the membrane has lost its integrity. Having then dark blue, stain and translucent cells indicate viable cells. Since these cells membranes are intact that prevent the entrance of the dye, the cells have the ability to continue reproducing and performing their metabolic functions. Given the observations made in this process, the majority of cells when stained intense blue evidence no viable diamond or dead cells. The yeast population is again deteriorated, which could affect the functionality in various applications such as bakeries, brewery, and biotechnology. Given the high rate and dye cells and could indicate to the yeast has been subject to adverse conditions such as inadequate temperature, desiccation, or exposure to toxic substances.
For the observation of epiphyseal cells with the help of the swain, a sample was taken from the inside and the cheek. In this case, it was mine. A small scraping was done of one of them. Subsequently, a transfer of the slice was made to the line, of which a drop of water and a drop of metallic blue were placed. Since the metallic blue piece along this line was splashed corner. The ten with the help and the strip on the swine, it was carefully placed of dot in slide of it, damaging it afterwards was covered with close slide and observed under the microscope and the loss was possible magnification to locate and epiphyseal cells. Here are the samples of the sheet of the epiphyseal cell with a drop of methylene blue and water. Here at 10x magnification you can see the cells in my mouth stained blue. There are some tiny pads, but there are also some that are quite well spread out and you can see in the moon. There you can see all cells perfectly.
Experiment we unloaded and 20% sodium chloride. Mounting the unloaded leaf under the microscope. Unload the cells before applying 20% sodium chloride. As a result, the application of 20% sodium chloride on the Lodian leaf, we can see that the chloroplasts are the direct to algal plant due to the loss of water, since the cells is releasing water from the vacuum to compensate the soil with a solvent.
For the observation of chromoplants, several slices were made on the tomato plant. The thinnest one was chosen and planted in this line. Additionally, a drop of water was added and it was observed under the microscope and different magnification for X and 10x in these organelles present in the tomato cell were observed, which are responsible to storing the pigments that give in color. In this case, the characteristic color of the tomato was in the color red.
The image showed a microscopic view of a heat-health relieving oval around the subepidermal and parenchymal cells. The cell halls appear whole definite, though stomata are not clearly visible. There might be chloroplasts or other inclusions. As we can observe, the potato in the macroscopic view shows that the tension generated in the small ovals visible within the cells walls of the amyloblast. This tension is created that the Lugol solution, which are used induce the tensions. Lugol solution helps facilitate the differentiation between the cell wall and the amyloblast found in this. In the lemon peel, we can observe that the chloroplasts are a bit more difficult to identify signs. Being a pale and the green and the color, the differentiation in it's not very noticeable. Whatever we can see that in the video, we can be identified more precisely. For the carrot, several cuts were made and the best one chosen, usually and did not too thick. It was mounted for a microscopic view where we can observe that the cell walls and the chloroplasts are very well organized, arranged in a brick-like pattern. Destaque different signs, no stain or the dye was applied. Additionally, the carotenoids can be observed which are responsible for the orange color. In the very collect on the university, we can observe that unlike the tomato, it has a slightly more intense red shading on the coloration. Its main characteristic responsible for this coloration is not entirely known, but under the microscopy, to the different cell walls present in the can be seen.
Conclusions. Viewing self and their components allowed us to better understand the basics of life. The use of dyes facilitates the observation of structures and the comparisons between animal and plant cells, highlights biological diversity. Furthermore, the evolution of cellular organizations from unicellular to multicellular demonstrates how cellular specializations has been key to the development of more complex organisms.